unlike most of the people you heard from today, I'm a wet bench biologist, and what I'm going to try and do is interest you in some of the problems we're interested in, many of which have led us to work with computational faculty in CB&B, including Hanyu Zhao and Jeff Townsend. So the general problem we've been working on for some years now is cancer, including especially cancer signaling pathways and DNA damage response signaling pathways. And the general notion is that fundamentally most cancers are driven by dysregulation of pathways that normally regulate growth of cell clones and embedding of cell clones within the tissue environment. So these are signaling systems that were developed really so that organisms such as your cells can have orderly three-dimensional structures. And we've known for some time that the cancer pathways are often involved in growth regulation, and that's led to the idea that all we needed to do to cure cancer would be to identify these pathways cancer by cancer on an individualized basis, and then have a collection of targeted drugs to either upregulate these pathways if they're shut off or interfere with these pathways if they are pathologically activated. And thanks to a lot of genomics and now epigenomics efforts and from people like Mark Gerstein and many others, it's been possible to analyze lots and lots of tumor specimens and infer the number of commonly activated oncogenic signaling pathways, and also connect them with gene mutations and epigenetic changes, so that working backwards by analyzing tumors or tumor cell lines in great detail, sometimes it's possible to predict not only describe which pathways are active, but also predict which pathways are amenable to interference with next generation targeted drugs that interfere with signaling pathways. So this kind of thing has been going on for about 15 years, which means we have a lot of clinical information on how well this works. And the good news is that these signaling and other targeted agents, when given to patients with the right tumor genotypes and phenotypes, they work. But unfortunately, they don't work for very long, typically over a period of months or years, and that I'm talking about a small number of years, say 3 years. Patients who initially respond very dramatically will become resistant. And so the biggest challenge for this kind of approach to cancer therapy is, in fact, you're looking at a moving target. How does this work? Well, at the pathway level, a simple example is in melanomas that have excessive ERK pathway activation. If you come in with an ERK inhibitor, and this is a receptor kinase activating the MEK kinase ERK pathway, this pathway intrinsically has a number of negative feedback loops. If you come in with a BRAF or ERK inhibitor, you interfere with these negative feedback circuits, and so overall the pathway downstream is reactivated. And this kind of resiliency signaling, which is just a homeostatic process, it's not just confined to that one pathway. There are often broad rewiring of signaling networks over a period of minutes, hours, and a small number of days when you throw in targeted inhibitors. So this is a dynamic process. A second kind of thing that happens is genetic or epigenetic resistance occurs by mutations that bypass the place where the drug comes in, or either by coming in downstream, a second mutation to reactivate a pathway downstream, or by activating a parallel pathway with the same consequences. A third issue that arose and is that even in a single cancer patient who develops resistance to a targeted drug, if you look at multiple metastases, you can find different genetic forms of resistance. So even at the genetic level, and certainly at the epigenetic level, these tumor clones are very plastic and very prone to deal with interdiction of signaling processes, especially with a single agent thrown in. Another practical issue for people dealing with these problems is not only are these tumor cell clones genetically plastic, but they are also phenotypically plastic, and especially through epigenetic changes, they can even jump cell differentiation lineages. So another common form of resistance is for tumor cells to adopt a very different kind of phenotype so that those cells are no longer responsive to the same kinds of therapies. Just by way of an example, here's a screen we did with a breast cancer cell line where we induced something called epithelial mesenchymal transition. This is a 41 by 41 matrix of drug combinations targeted at signaling molecules. Red means the cells are sensitive to these agents.
Excuse me, David, your slides are not progressing. We're still in the first one.
Oh, well, thanks, Lisa. They're progressing over here.
It still says just the Stern Lab and signal transaction and cancer.
Okay, I'm going to reshare my screen. I'm seeing a very nice progressive.
I mean, we can hear you, but we're not seeing the screens.
Yeah, well, I'm glad you piped up. So again, let me just redo the screen share and see if that helps.
Okay, sorry about that.
Okay, now I'm no longer seeing the option to, oh, here we go. How's that?
There you go, perfect.
Okay, so I'll keep going because we're short on time. So this is an example, though, where drug combinations targeted different pathways. Control cells are very resistant to are very sensitive to these drug combinations that's shown in red, but if you induce epithelial mesenchymal transition, they become very resistant, which is green. Okay, another final problem is that tumor cells are heterogeneous and tumors consist not only of the clone of tumor cells, but surrounding cells such as fibroblasts, fat cells, vascular cells, and especially immune and inflammatory cells, which we now know are extremely important because many solid tumors are partly controlled by a weak host anti-tumor immune response that can be ramped up with the appropriate immune modulation drugs. So these cancer problems create a series of systems level problems that I hope all of you will consider working on. And just in terms of understanding how normal signaling works and how cancer signaling works, we basically know the point-to-point wiring diagrams of many major signaling pathways. We know general classes of processes that are regulated by these pathways, and we know some kinds of mutations that are recurrent in cancer that activate or inactivate these pathways, leading to disease. But even though we have a sparse map of signaling connectivity, it does not begin to capture the range of connections among these molecules. For example, the connections of protein kinases with their substrates. The modification of regulation of most signaling proteins is incompletely understood. Signaling complexes involve multiple protein-protein interactions. Signaling complexes function at a local level intracellularly, so accounts where they are located. And these pathways are connected in complex interregulated networks. So the kind of things that Dr. Gerstein has been doing with transcriptional networks are repeated in spades in protein networks. And for technical reasons, they are more difficult to evaluate. The bottom line is that we cannot yet predict static or dynamic changes in information flow through these networks. And as a consequence, we can't really deduce the activity of the signaling system of a network based either on protein measurements or on transcriptional measurements. So being able to do this, though, would be a great goal, because when we obtain tumor tissue from patients, we can measure genomics very completely and accurately. We can measure epigenomics and transcription profiles, again, in a comprehensive way. So if it becomes possible to infer the static and predicted response activities of signaling networks based on DNA sequencing, DNA copy number, and transcriptional analysis, that would make it much more likely that we would be able to predict how these pathways will respond initially and over a period of time to signaling interventions. So in our lab, we are interested in these processes. Again, the most important, a great big challenge for all of you would be to try and develop predictive measures for drug response based on transcription profile, which is the most complete phenotypic measurement we can make of a tumor. And also to understand how intercellular interactions function in modulating their phenotypes. In the last minute, I'd like to just promote the Cancer Biology Training Program, which I lead. This is a program to provide cancer biology training and genetics training, and also to provide clinical exposure to practical clinical issues in oncology for people like you. The program includes clinical mentoring, some coursework above and beyond your CB&B requirements, participation in tumor boards and Yale Cancer Center activities. If you're interested, just look on the BVS website. There's information, or contact me. This is a program several CB&B students have participated in and are participating in, and applications for first-year students are in the spring. So thank you, and I guess we can take one or two questions.